We really appreciate this opportunity to talk with you all today about the work we've been doing on spatial occupancy models, and then our SP occupancy R package as well. And I will apologize in advance. I'm a bit sick, so I might be taking a few more drinks of my water than I might usually. So just a heads up there. So first, just an overview of what the presentation is going to be today and what the webinar will be. And first, just to make sure we're all on the same page, just going to talk, kind of give a brief overview of occupancy modeling as well as spatial modeling, and then really spend the bulk of the time talking about implementing these types of models in SP occupancy, going through the functionality SP occupancy supports, as well as the syntax, and going through a couple of examples. We're going to spend most of the time talking about a single species example, but we will get a bit into a multi-species model as well if we have time. And then, of course, there will be plenty of time for Q&A at the end. And I will just mention, so I'm just going to zoom quickly over to the GitHub page just to mention how you can download this. So here I'm on the GitHub page for the Stat Seminar Series. If you just want to download everything as a zip file, you can click on code here and then download everything as a zip. That's going to download all of the materials from past seminars as well. And then you can just access and navigate to our specific folder where we have the resources we use today. So first, just kind of thinking really broadly as to why we want to implement an occupancy model. An occupancy model is a specific form of a species distribution model, which has numerous applications across both applied ecology, conservation, as well as theoretical ecology. And kind of the basic questions we're trying to answer with species distribution models is where do species occur? How does this change over time? And then what drives these variations in where species occur across space, and then also how that changes over time. All right, and it sounds like a really straightforward task, but of course, if you've ever worked with data and tried to fit a species distribution model, you know that there's a lot of complexities that arise when you go to try to fit one of these models. Okay, and kind of two major complexities that we're going to talk about today, and that we account for in SP occupancy, is imperfect detection and then spatial correlation. And so the concept of imperfect detection is relatively intuitive. So you can imagine if we're going out to do some sort of survey here, we're doing a point count survey of birds, we're going to try to count all the species that we observe in an area. Okay, and what we'll do is we'll be able to successfully identify a lot of those species. But then what's going to happen is we're just going to miss some species when we go to count them. So for example, these two individuals down here, we miss them when we go to do the survey. And so this process where our detection of these birds is not perfect, or it's imperfect, can lead to biases if we try to fit a species distribution model without accounting for this imperfect detection process. Okay, and of course that lends to the question of how exactly do we go about accounting for imperfect detection, and that's where occupancy modeling comes in, which is kind of the go-to way for accounting for imperfect detection when modeling kind of detection or non-detection data. Okay, and the basic concept of an occupancy model is that you're going to perform multiple surveys or visits or replicate surveys at each site that you're interested in. Okay, and what this allows us to do is it allows us to separately estimate occupancy probability from detection probability. Okay, and the distinction between those two is occupancy probability is really what's the probability a species occurs at this location, whereas detection probability is what's the probability I detect this species given that it's actually present at that location. Okay, and the basic data structure that we're going to work with in occupancy modeling is kind of this detection, non-detection data matrix that I have shown here on the right. Okay, and so basically what this matrix consists of is the rows correspond to the sites in your surveys, and then the columns correspond to those repeat visits or those repeat surveys at each site. Okay, and what each cell in the matrix is it denotes whether or not you observed your species of interest at that site during that given survey. Okay, And one thing I will note here is that you can, of course, have situations where you're missing some surveys at a given site, and that's totally fine to do in an occupancy modeling framework. You can have missing data in your modeling framework. So now that we know kind of what the data looks like, how do we actually implement it in an occupancy model? What actually is an occupancy model? Okay, it's really a relatively straightforward extension of a logistic regression or a generalized linear model. Okay, so what it is essentially kind of two logistic regressions placed on top of each other, where the first logistic regression models occupancy probability as a function of site level covariates. So where does that species occur, and what covariates kind of drive where that species occurs. And then the second is where we model detection probability. So the probability we detect a species given that the species actually occupies the site, okay? And this detection probability can vary not only across space, so it can vary with spatial covariates, but it can also vary across the different surveys as well, okay? So we can model both site and survey level covariates in a model for detection probability. And a key assumption we make in a standard occupancy model is that you can only detect a species if it truly occupies a site, okay? So in other words, we assume that there's no false positives. You don't observe a species at a location where it actually does not observe, okay? Now, taking those kind of large scale view and then putting it in kind of equation format, what does that exactly look like? Well, we can imagine that we sample a total of j sites, where we use lowercase j to index across the sites. And then at each site, we have k sub j replicates, and lowercase k is used to index across the replicates. Okay, so our ecological model, or ecological submodel that we're most interested in is that occupancy portion of the model. Okay, and so what we have is this true presence or absence of the species at each site j. So we denote that as z sub j. We model this as arising from a Bernoulli distribution. And what we're really interested in is this occupancy probability, psi sub j, which we model using a kind of logistic regression framework where we can include a variety of site level covariates that influence, or we think will influence, occupancy probability. And then the second model, the detection or the observation submodel, is kind of linked to this occupancy model by assuming that we only detect individuals at sites where the species actually occurs, okay? So the data that we observe are denoted as y, so y sub jk is the detection or the non-detection of our species at site j during replicate survey k, okay? And what we assume is this arises from Bernoulli distribution, but this is conditional on actually observing the species, or actually the species being present at the site, okay? So notice that z sub j is in this equation. So if the species doesn't actually occur at the site, we can't detect it. Again, no false positives. And this p sub jk is the detection probability, which varies across both site and survey level covariates. All right, so that's the basic single species occupancy model. Of course, we can be interested in multi-species models as well, where we have a community of species that are interested in modeling. And in SP occupancy, we have kind of two different flavors of multi-species occupancy models. And if you're familiar with multi-species occupancy models, you'll know that there's a whole suite of different flavors that exists in the literature. And so what we have in SP occupancy are kind of the D'Orazio-Royal multi-species occupancy model, also known as a community occupancy model, where we treat species as random effects arising from a common community level distribution. And then we also have joint species distribution models that account for imperfect detection. So multi-species occupancy model that allows for species correlations. And we account for this using a factor modeling approach. And I've linked to our preprint there, which goes much more in depth on these models and how they can be useful. And kind of the key concept of a multi-species occupancy model is, again, thinking what the data looks like. And what we can imagine is taking that detection on detection data matrix for each species, and we just now have a separate matrix for each species that we're interested in. Okay, so this top layer is our first species, the second layer is our second species, our third species, and our fourth species, and so on for all species in our community. And thinking about it this way is going to be really important for how we structure the data for multi-species occupancy model and SP occupancy. So keep this figure in mind later for when we go through the code. All right, so the occupancy modeling addresses the imperfect detection component, but what about that spatial autocorrelation that I mentioned, that additional complexity that we want to account for in species distribution models? Well, spatial autocorrelation in its most simple form is just saying that things closer together in space tend to be more similar than things that are farther apart in space. Okay, so a really simple concept that seems to apply to a lot of things in ecology. Okay, and it's important to think about what leads to the spatial autocorrelation in species distributions. All right, so we can imagine this comes from kind of environmental drivers or the habitat requirements of a different species. Or it could come from more biotic factors, so things like the dispersal capabilities of your species of interest or different sorts of biotic interactions. And our initial approach to accounting for spatial variability and where a species occurs is to try to explain that variation using covariance. So things like forest cover or different climatic variables like temperature or elevation, so different variables that you think will influence where a species occurs over space. But when spatial autocorrelation becomes a problem is when spatial correlation exists after you account for those spatial covariates. Okay, so this is called residual spatial autocorrelation. And residual spatial autocorrelation can arise from a variety of processes, but perhaps most common as a result from missing or unavailable covariates. Okay, and so we can imagine if we're modeling a species distribution across a large spatial area, that we have kind of broad scale spatial covariates, but we don't have those really fine scale spatial covariates that really help determine where in a landscape a species is using that space. Okay, and like imperfect detection, if we don't account for spatial autocorrelation, then this can lead to bias in our estimates of a species distribution. All right, and so the way we account for spatial autocorrelation is to use spatial random effects. All right, kind of at a high level, what a spatial random effect is that each site has this kind of local adjustment in occupancy probability. Okay, what I mean by that is that at each site J, basically based on the spatial structure of the data, we're going to adjust that occupancy probability to be higher or lower depending on kind of what the spatial autocorrelation looks like in the data. Okay, and this involves kind of, for the model covariance functions that we use in SP occupancy, generally two parameters, so a spatial variance parameter and a spatial decay parameter, which I'll talk about a bit in the next slide. And I will just mention really briefly, not going to go into a detail, but this including spatial random effects doesn't come at no cost. So there is this concept of spatial confounding that we should be aware of. And what I mean by that is that if you include spatial random effects in your model, what can happen is that the spatial random effect is actually correlated with the spatial covariance in your model. Okay, and when that occurs, sometimes you have to be careful when you interpret the regression coefficients or the effects of the covariance that you include in the model. Okay, and I've linked a paper by Hanks et al. In 2015 that goes into that concept much more in depth. So what does a spatial occupancy model look like compared to a single species occupancy model? Well, it's really a simple extension. So we have our detection submodel that stays exactly the same. And what we do change is in the occupancy submodel, where we incorporate, again, that kind of local site adjustment, that spatial random effect to each site J. And we denote these using the W. So W sub J is our spatial random effect. And this W sub J is assumed to arise from a multivariate normal distribution or a Gaussian process, which is governed by a spatial covariance matrix sigma. Okay, and the way we model these random effects in that way I just mentioned is using a Gaussian process, which is pretty much the gold standard for modeling spatial data. And really what is nice about a Gaussian process is just it's extremely flexible and allows us to account for a variety of kind of spatial structures in our residuals, okay? And so really what we specify is a spatial correlation function, which helps determine the structure of our model, okay? And what I have shown here are kind of the different forms and of what that spatial random effect can look like in relation to the estimates of the parameters that we're estimating, okay? So this sigma squared parameter is the spatial variance parameter that we estimate. which controls kind of the magnitude of those spatial random effects, where when its value is low, there's kind of little variability in these effects across space, but then when it's high, there's much larger variability in the magnitude of the spatial random effects. And then our second parameter is this phi, which is a spatial decay parameter, which essentially controls kind of the smoothness of that spatial process. So as phi decreases, it becomes smaller. We see that our spatial process becomes much more smooth over space. Okay, so these parameters kind of control what that spatial correlation looks like, and are the parameters that we estimate in SP occupancy. All right, so Gaussian processes have been around for a super long time. So kind of what's the downside of using them? Well, they're super computationally intensive for big data, even moderately sized data with a couple hundred locations. And so what we do in SP occupancy is we use an approach called nearest neighbor Gaussian processes, which I'm not going to go into any detail on, but you can check out the Data et al. 2016 paper for all the details on that. And then our implementation in SP occupancy in our paper there. But this is an approach that allows us to account for spatial autocorrelation in a much more computationally and efficient manner than using a full Gaussian process. Okay, so that leads us right into SP occupancy. That last slide kind of gives the motivation for developing SP occupancy, is to develop a set of efficient models and software that can accommodate both spatial autocorrelation and imperfect detection when trying to fit species distribution model. Okay, we do this in a Bayesian framework. And perhaps one of the nice things about SP occupancy is the workflow is completely in R. So you're using standard R model syntax. So in order to fit these models, you don't have to turn to Bayesian programming languages and kind of fine tune those approaches and kind of learn those programming languages in order to fit these types of models. And at the moment, there's kind of 4 key kind of occupancy models that SP occupancy supports. So we have our single species and our multi-species occupancy models that I mentioned before. And then we also have functionality to account for data integration, so integrating multiple detection, non-detection data sets in a single model. And then more recently, I've incorporated some multi-season or some spatio-temporal models to estimate kind of trends in occupancy over time while accounting for spatial autocorrelation. Okay, I'm going to talk primarily about these first 2 today, and then there's also examples on the other 2 in the GitHub archive. Okay, so why Bayesian for occupancy modeling? I just wanted to mention this really quickly, that we're fitting these models in a Bayesian framework, and there's a lot of flexibility that a Bayesian framework affords us. You know, if you're familiar with occupancy modeling, you know you can fit maximum likelihood occupancy models using unmarked. But the Bayesian approach allows for additional flexibility to account for spatial autocorrelation, allows us to extend to these multi-species frameworks or potentially use multiple data sets, as well as getting full uncertainty propagation for our estimates, which can be really important in communicating our results to stakeholders. So the workflow in SP occupancy kind of has these 6 steps. So the first step would be kind of data simulation if you're working with simulated data or preparing the data for the format that SP occupancy requires it to be in. And I think this data preparation point is probably the trickiest part for fitting a model in SP occupancy. So I'm going to spend a good amount of time talking about that in a second. And then kind of the cool part of the package is when you go to fit the models. And then of course, after you fit a model, you want to validate it. You want to compare it to other models. And then you're going to summarize the results as well as do different types of prediction to generate species distribution maps and other posterior quantities that you're interested in. Okay, so our example data set today comes from the Switzerland Breeding Bird Survey in 2014. So if you've read the applied hierarchical modeling books, you might be familiar with this data set. It's commonly used as an example in those books. And what this data consists of is replicated detection on detection data from 266 survey locations that are distributed throughout the country of Switzerland. And what I'm going to talk about primarily is a single species model that works with the European goldfinch, as well as a multi-species model working with 20 species from the subset of this community. And our goal is going to be to produce this map, or this figure that I have here, where we map occupancy probability as a species distribution map And then also take a look at that random spatial effect that we estimate as well to see how it varies across space. And before we get into the code, I do just want to mention the GitHub archive has a bunch of different examples. So for fitting single species and multi-species models using an amphibian data set, example of an integrated model using some bottlenose dolphin data from Lev et al. In 2021. And then a multi-season occupancy model as well. So if you're interested in those types of models, I encourage you to check out those resources we have there. Okay, and just to mention before I get over to the code, what we are working on right now should be up in the next couple of months is incorporating spatially varying coefficient models into the package, okay, where we actually allow the regression coefficients to vary over space. So that should hopefully be up by the end of the year, so keep your eyes out for the update. And then we're also working on some multi-species extensions for spatiotemporal models, as well as multi-species integrated models as well. Okay, this is just a slide just to mention all the resources. You can check those out in your own time. I do post all the package updates on Twitter, so give me a follow if you want to keep an eye out for new functionality. And just lastly, before I zoom over to RStudio, we'll acknowledge my co-authors on some or all of this work, Lee Sipkin, Mark Harry, and Sudipta Banerjee. They've helped out in different forums for developing the models and the package itself. Okay. All right, so with that, we'll zoom and head over to RStudio for the code we're going to work with. So I'm going to go ahead and switch my screen. If you want to follow along, you can open up the code as well. I'm going to close these out first, actually, just to show you where these are located. So the two scripts we're going to work with today are have to do with that Swiss breeding bird data that I mentioned. Okay, so if you navigate to the code directory inside of the GitHub archive, you'll see all the examples that I mentioned. And what we're going to work with are the two that start with Swiss MHB. Okay, so the single species data set as well as the joint species distribution data set. Okay, so you can open up these two scripts. And we're going to spend most of our time talking about the single species example. Okay, so open up that script if you're following along. And I will mention, you might see my RStudio is slightly different from the default. I have my script window on the left over here, and my console window on the right. Okay, so just to give you a heads up on that in case you were wondering what was going on. Okay, so the first thing I'm going to do, I'm just going to clear out the work directory just to make sure we have make sure we have a clear space. I'm going to load the package. There's a few other packages that I used to kind of summarize some of the results and make the species distribution map. So the first is MCMC-Vis for kind of summarizing the MCMC results using some quick code. And then a bunch of packages for making the species distribution map. Okay, so you can load those if you want to generate the maps on your own machine. And I will mention in the GitHub archive, you should see a RStudio project file. Okay, so it should be this SP occupancy examples, RStudio project. So I encourage you to open that up because that's going to automatically set your working directory to the kind of home location of the folder. But if you don't use the RStudio project, then you're going to have to run this line of code here to set your working directory to kind of the home directory of our code for this webinar instead of the location where the script is located. Okay, and then the last thing of kind of housekeeping before we get started is just to set the seed so we each get the same results when we go to fit the model. Okay. And again, our example with this code is we're going to fit a single species occupancy model. We're going to fit a single species non-spatial and spatial occupancy model to generate a species distribution map of European goldfinch throughout Switzerland. Okay. And while I'm not going to spend much time talking about the other models in SP occupancy, the kind of basic arguments are really similar across fitting a single species model versus a multi-species model versus a spatiotemporal model. So a lot of what we'll talk about today applies to those kind of more complex models as well. All right, so the first step, I kind of have the sections delineated by the different SP occupancy functionality, is this data prep section, okay, which I think might be the trickiest part, which I get a lot of questions on, is organizing the data for fitting SP occupancy, or fitting models in SP occupancy. Okay, and so what we're going to do is we're going to load this object, this R data file that's in the data directory. We're just going to bring in this object called data.goldfinch. Okay, so if I go ahead and run that, we should see this data.goldfinch object be loaded into our environment. And so let's take a couple of minutes to talk about what exactly that structure is and how we need to structure our data for fitting models in SPI. Okay, so if I go over here, what we see is that this data.goldfinch is a list of 4 objects. Okay, and it's this format, this list format that's required for all SPI occupancy functions. Right? So the first object in the list is labeled as Y. All right, and what Y is it's the actual detection, non-detection data that you obtain. Okay, so if you recall from the slides that matrix where our rows corresponded to sites, and then our columns correspond to those repeated visits, those multiple observations, that's exactly what we have in this Y object. All right, so it's a 2-dimensional matrix or data frame where those rows correspond to sites and columns correspond to repeat visits. All right. And if I go ahead and actually take a look at this object, data.goldfinch, and take a look at Y, what I want to point out is that missing values are included in this using NAs. So if you have an uneven number of replicates at each of your sites, then what you need to do is put NAs for those site replicate combinations that we don't have any data for. Okay, and SPI occupancy will just kind of filter out those NA values. All right, let me rerun this so we can look at the rest of the objects. And the second component of the list is labeled as OCC.cov. So this is the occupancy covariance, the covariance you want to include to model occupancy probability. All right. And how these are stored is a data frame or a matrix where the rows correspond to the number of sites you have. So here we have 266 sites. And then the columns correspond to the different covariates that you have in your model. Okay, so in this case, we're going to fit a model with elevation and percentage forest cover in the area. Okay. Now, where things get a bit tricky is including the detection covariance in the model. So the covariates that you think are going to influence the probability you detect the species, given that the species actually occurs there. Okay. And this is contained in the object det.covs. So that's another component of this list. And det.covs is actually a list itself. Okay. So it's a list itself where each element of the list is a covariate that you want to include on detection probability. Okay. And the reason why this is a list is because these covariates on detection can vary not only across space, across the different sites, but also across those different surveys as well. Okay, so each of these that we have included here are covariates that vary across both space and the multiple visits. All right, so here we have date, so the date of the survey, as well as DUR, which is the duration of the survey. Okay. And if you had a covariate on detection that only varied across space, then you could just include this as an element of det.covs as a vector of in this case, 266 values, or corresponding to the number of sites you have. Okay. And then the last component is this coords, which is a 2-dimensional matrix containing the spatial coordinates of each of your sites, okay, where the columns correspond to the X and Y values of those coordinates. And what I will mention here is that it's kind of 2 things. First, this isn't actually required for fitting a non-spatial occupancy model, okay, but if you want to account for spatial correlation, you have to include this in the model. And then I'll also mention we assume that the coordinates are in a projected coordinate system. So that's just something to keep in mind when you go to fit these models. Okay. So I know that was probably a bit dry talking through that formatting of the data object, but I think that's a really crucial part for fitting these models in SPI occupancy. All right. So the next thing we'll do before we go to fit the model is just take a look really quickly at the spatial locations, just to get an idea of our sites. I think I'm going to have to make that a little bigger and then we get this to work. And if we take a look at our survey locations, just with this simple plot, if you're familiar with the shape of Switzerland, you'll see that our points kind of are evenly distributed across the country, following this standard grid. And our goal is to predict occupancy probability across the country. Okay. All right. So next up, now that we have the data in the format, it's prepared in the format we need for fitting an occupancy model, we can actually go ahead and fit the models. All right. So first thing we're going to do is we're going to fit a non-spatial single species occupancy model. And the name for fitting this model is PGOCC or PGOC, which stands for Polyagamma Occupancy Model. And that polyagamma is a statistical technique that we use to kind of improve the efficiency of these types of models. And so this PGOC, this is kind of the base name for all model fitting functions in SP occupancy. So all the names are going to be kind of extensions of PGOC. And there's a bunch of arguments that you can specify to PGOC, which kind of keep showing up as I accidentally hover over it. But I'll kind of just talk through the main ones that we need to get going and to fit the model. All right, so the first is the OCC.formula. So the formula for the occupancy portion of the model. And this is where you're going to include those covariates that you think are going to affect occupancy probability. All right, and so what we have here is we specify it using standard R model syntax. All right, so the only thing I'll note is that unlike when you fit a model with, LM or GLM, you have a left side and a right-hand side of your model formula. Here, this is just the right-hand side. Okay, so you're going to start it off with the tilde, and then you're going to include the covariates that you want on your model. All right, so in this case, I have a linear effect of elevation, a quadratic effect of elevation, as well as a linear effect of forest cover. Okay, and I will point out that notice I can use the scale function to standardize my covariates to have a mean of 0 and a standard deviation of 1, which is often useful for fitting models in the Bayesian framework, in particular when they're on really different scales. Okay, so the next thing we'll do is we're going to specify the detection formula. So that's the det.formula argument. Again, this is a formula using our kind of standard R model syntax where you specify the right-hand side of the formula. And here what we do is we include a linear effective date, a quadratic effective date, as well as a linear effect of duration. All right. Then you have to specify the data. So in this data argument, you're just going to send in that list with those 4 components that I mentioned before. And then the rest of these arguments I have specified here have to do with the Bayesian approach we use to fit them. Okay, so we use Markov chain Monte Carlo or MCMC to fit these models. I'm not going to go into details on that approach if you're not familiar with it, but you can check out all the package website materials for more information on that. But basically, we're going to run the model for 5,000 samples for kind of 3 chains, 5,000 samples for each of 3 chains. Okay, and lastly, this n.report argument controls kind of how often we report information to the screen. Okay, so I'm going to go ahead and run this now that I've kind of gone through the basic arguments. We should see on the right, the model should fit really fast. This is a single species occupancy model without any spatial correlation. So it's the simplest model we can fit in SP occupancy, so it's going to be quite fast. But if I scroll up to the top of it, what I will mention is kind of the output that comes from this. All right, so first thing that shows is this preparing to run the model section. And what's included here is kind of messages to let you know of any assumptions you're making when you fit the model. Okay, so if you're familiar with Bayesian models, you'll notice I didn't say anything about initial values or priors. Okay, and so those are arguments that you can specify to PGOC, or you can use the default values that PGOC provides, which it's going to tell you in this section what those values are. Okay, and then we have our model description section that just kind of gives you information about the arguments that you use to specify the model, some information on the number of threads you use to fit the model. Okay, I will mention that fitting the model with multiple threads is only going to lead to speedups for the spatially explicit models. All right, so for the non-spatial models, it's really not going to make any difference. But for the spatial models, it can make some significant improvements in computational runtime. Okay, and then you have this section that prints out progress of the model, which in this case wasn't all that useful because the model ran so fast, but generally that'll be some useful information we have. Okay, and really the first thing I do after I fit a model in SP occupancy is I use the summary function to get kind of a quick understanding of the results that come kind of the estimates and the results from the model. All right, and so you just do summary and then out. So out is what I call the model name. Excuse me. And so if I run that, you can ignore those messages. Those just have to do with the graphics state. What you should see here is some information presented on the model fit-in, or the arguments used to specify the model, as well as information on the model, how long it took. So in this case, it ran super fast, much less than a minute. And kind of the main information we're interested in is in these two tables where we have the occurrence regression coefficient estimates, as well as the detection regression coefficient estimates, okay? And so there's kind of printed a bunch of different information that we have here, kind of the mean of your posterior estimate, standard deviation. These 2.5 and 97.5 quantiles can be used to form a 95% credible interval. And then you also have the posterior median as well. And then these last two columns relate to assessing convergence of the Bayesian model. Generally, we want these R-hat values to be less than 1.1, which we see is the case for all these parameters here. All right, so if we just wanted to kind of quickly interpret some of these results, we can see a negative effect of both linear and quadratic elevation. If we take a look at the credible intervals for that, we have negative 1.89, negative 0.73, negative 1.76, negative 0.89. So they don't include 0, which indicates that's a pretty important effect. Then we also see a negative effect of forest cover on occupancy probability as well. Okay, and you could do the same thing for the detection covariance, interpret them in the same way. All right, so we fit a non-spatial model, and as I mentioned, kind of all the models in SP occupancy have that kind of same basic structure for fitting the model. So now that we're familiar with the single species model, the extensions are going to be pretty straightforward to the rest of the models. All right, so if we try to fit a spatial single species occupancy model, we do that using the function SPPGOC. Okay, so the SP before it just kind of emphasizes that it's spatially explicit. And the arguments for SPPGOC are really similar to what we saw with PGOC. The main difference is probably in how you have to specify the number of MCMC samples. Okay, so in this case, what we do is we split the total number of samples up into a series of batches, where each batch is comprised of a set number of MCMC samples. Okay. And so to specify that, we have, what you do is you specify the total number of MCMC sample batches, which is this n dot batch, as well as the length of each batch. So how many MCMC samples are in each batch? Okay, so here we set that to 25. And the reason we do this here, we make it a bit more complicated, is because we use what's called an adaptive sampler for estimating some of the spatial parameters. And so that requires us to specify these arguments, all right? But if you want to figure out the total number of samples you're running for, you would just do n dot batch, which in this case is 400 times the length of each batch, which is 25, which in this case is 10,000. Okay, so we're running 10,000 MCMC samples. And then the next two arguments are the two arguments we're going to specify to fit a spatial model. So this NNGP argument controls whether or not we use a nearest neighbor Gaussian process or a full Gaussian process. As I mentioned before, the NNGP is going to be way faster and give very similar estimates to the full Gaussian process. And when you fit an NNGP model, you have to specify the number of neighbors. The default value for this is 15, which is shown by Andy and his colleagues to be really robust for pretty much any sort of data set. But here I've set it a bit lower to 5 because it helps speed the models up even more. And I've also played around with this data set a bit, and 5 seems to do quite well. Okay, so if I go to run this model, we should see that now the spatial occupancy model runs kind of slow enough that this information on our model progress is relatively useful to us because it doesn't just go super fast. And what I'll mention that you'll kind of see here is being printed these acceptance rates and these tuning variances for one of our model parameters. So parameter phi, which is that spatial decay parameter. It basically what I'll mention is that you want to make sure that these acceptance values are not super close to 100 or not super close to 0. All right, you kind of want them to fall somewhere in the 40s or the 50s or something like that. And if you're seeing a case where it's constantly 100 or constantly 0, what you might have to do is change some of the initial values when you go to fit the model, or you might also have to change the tuning variances, the initial tuning variances. And to do that, you can check out the manual page for SPPGOC for how to include those arguments in the model. Okay, but here we see they're kind of hovering around 40s, 60s, so I think we're pretty good to go. And then again, I'm going to take a look at the summary to see what the kind of get a basic summary of the results. I'm going to make my screen a bit bigger so it prints out nicer. Get rid of that warning. There we go. Okay, so if I take a look at the model summary, we see the model, the spatially explicit occupancy model with 266 sites fit in less than a minute, which is pretty good. And if we look at the summary, it's really similar to what we saw with the non-spatial model, where you have your kind of coefficient estimates and the different criteria for those. I will mention some of the R-hat values and the effective sample sizes are not ideal, so we probably want to run this a bit longer, but generally it's good for kind of our purposes right now. And then you will also see the estimates of the spatial covariance parameters, that spatial variance sigma squared, and then that spatial decay parameter phi, where that's the value of our spatial variance parameters is pretty large at 1.72, so that suggests that there's pretty substantial spatial variability in our model. Okay. All right, so we fit these two models. Now we can our next step would be to assess the goodness of fit of these models. I'm not going to spend much time on this at all because I'm kind of going a bit slower than I originally thought, but basically what we have is the function PPC OCC, which stands for posterior predictive check for an occupancy model, allows us to do numerous forms of goodness of fit tests for SP occupancy model objects. Okay, so here what I'm going to do is I'm going to do a posterior predictive check for my non-spatial occupancy model, as well as my spatial occupancy model. And these should run relatively quick for these simple models. And these resulting objects, so ppc.out and ppc.out.sp, will have a variety of information, variety of objects inside of them for assessing goodness of fit, which you can explore more in depth. But if you kind of want a quick summary of goodness of fit, you can calculate a Bayesian P-value using this summary function. So if I calculate a Bayesian P-value for my non-spatial model as well as my spatial model, what we'll see is Bayesian p-values of around 0.2 for both of these. And for Bayesian p-value as a measure of goodness of fit, we generally want it to fall kind of close to 0.5. And we come up with these kind of arbitrary cutoffs of 0.1 and 0.9, which indicate kind of bad model fit. So here we're at about 0.2. So our model fit, we can assume is fairly adequate for our purposes here. Okay, and then the next step we could do would be to compare models. All right, and so we can do that using WAIC as an information criterion approach. All right, so the function to do that in SP occupancy is to use WAIC OCC. All right, and so we can calculate the WAIC for both the non-spatial and the spatial model. Okay, and if we look at these, lower values of WAIC correspond to better model fit, similar to what we think about with the AIC. Here we see that the value for the spatial model is lower than for the non-spatial model. So we'd probably continue our analysis using the spatially explicit model. Okay, so that's what we're going to do going forward. We're just going to work with the spatial model. All right. And so now if we want to kind of summarize the results from the model a bit more in depth, as I've mentioned numerous times before, the summary function will be your best friend for getting some kind of initial and quick interpretations of the results. You can also take a look at the actual objects in the model object that comes from when you fit an occupancy model. So there's a whole lot of stuff in here, most of which you won't really need to pay attention to. But if you want to actually extract the posterior samples for the coefficients or the parameters you're interested in, all of the objects that have samples at the end of them are those posterior samples. Okay, so you can take a look at those and kind of summarize them in any way you want to. These samples objects are all stored as MCMC objects from the CODA package. Okay, so any sort of summary that you can do with CODA objects, you can do the same thing with these posterior samples. All right, and so that kind of leads right into some kind of summaries, some quick summaries with the MCMs of this package. Since we set these as CODA objects, we can kind of make a quick plot of our results. And kind of pop this open. So what we can see here are kind of summaries of the occupancy coefficients. All right, so we see that negative linear quadratic effect of elevation, as well as a negative linear effect of forest cover. Okay, we see that none of these overlap with 0, indicating that they're pretty important covariates for determining the occupancy probability of the species. Okay, and then you can also do the same thing with the detection covariance or kind of any other parameter that you want to fit. So here I have shown the kind of effects we estimated on the detection portion of the model, where we see kind of a moderate effect of duration, as well as a moderate effect of kind of quadratic data as well. Okay, so the last thing I'll talk about is prediction. All right, so how we can predict occupancy probability across Switzerland and generate a prediction map for this data. All right, so to do that, we're going to load a couple more objects that are contained in that data folder. And what this is going to do is going to load 2 objects. So the first will be coords.0. So if we take a look at coords.0, just using str, what we'll see is that we're going to predict at about 42,000 new locations. And these new locations are kind of a gridded, kind of a grid across of Switzerland to generate a clean map. Okay. And then if we take a look at the pred.swiss object, which was also contained in that object that we loaded, what we'll see is this is a matrix with rows corresponding to sites, and then columns corresponding to our 2 covariants that we have in our model, elevation and forest cover. Okay. And one of the important things to remember is if you standardize the covariate values when you fit the model, when you go to do prediction, you have to standardize the values, the covariates, by those same values used to fit the model. Okay, and so that's what these few lines of codes here does to generate this elevation.0 and forest.0 vectors does. So it's going to generate those covariate values on the scale that we use to fit the model. Okay. And then the object that's required by SP occupancy for prediction is you have to generate a prediction design matrix. Okay, and so what that is just a column with, or a matrix with rows corresponding to the sites that you want to predict at. And then each column corresponds to one of the covariates that you use to fit the model. Okay. And one of the trickier parts about this is that you will need to include, if you fit the model with an intercept on occupancy probability, you're going to need to include a column of 1s in that design matrix. Okay, so make sure you don't forget to do that if you're going to do prediction. And then otherwise, you just include the covariates you use to fit the model. Okay, so here I have our elevation values, I have our elevation squared values, and then I have our forest cover values. Okay, and I will mention that the order you put these into this matrix should match the order that you specified the covariates in the OCC.formula portion of your occupancy model fit. Okay, and I'm just going to run this just to get the prediction going, because it's going to take a minute or two since we're predicting over about 42,000 sites. But basically, to do this prediction, what we have is you can just specify this predict function. What you send in is the occupancy model object, so this out.sp. You send in that design matrix, and then for spatially explicit models, you'll also need to include that coords.0 object. Okay. And while this is running, we'll see this kind of output. So similar to when we fit the models, you have kind of information being shown for predicting across these new locations. I will note something that might be a bit confusing is here it's showing that we're predicting at 42,009 new locations. And if we're paying super close attention, the prediction objects we had about 42,200 something sites. All right. So the reason it's showing 42,000 is because these are the new sites that we're predicting at. Okay, so for the sites that we actually already have occupancy probability generated at, we already have those values, so we don't need to kind of do a double prediction. Okay, and I will mention, so if we just take a look at the help page for predict while this is running, and I actually already have it open, but if you wanted to search it, I will just mention sometimes it's a bit tricky to find these help pages for the predict functions, right? So for SP occupancy model objects, if you want to search the help page for a prediction function, what you can do is type in predict dot and then the name of the SP occupancy model function that you use to fit the model. All right, so in this case, fit a model with SPPGOC. So you type predict.SPPGOC, and that should load up the help page. Okay. And a couple of things I'll mention here is, as I've done throughout, there's a bunch of other arguments to the functions that I've kind of ignored. In particular for predict, here we're predicting occupancy probability to generate a species distribution map for our species of interest. But you can also change this type argument to predict detection probability if that's something you're interested in. If you wanted to predict detection probability, that can potentially be helpful depending on your study objectives, especially if you're trying to help design future surveys as well. You can kind of generate predictions of detection probability to figure out when it might be most optimal to go out and perform your sample surveys. Okay, and then while this is running again, I'm going to open up the help page for SPPGOC. So we have about 20% of the prediction to keep going. And so one thing I will mention here is, in this case, we're kind of this arbitrary example. I'm mostly interested in predicting, generating the species distribution map. Okay, in that case, it might be useful to do model comparison using kind of an assessment of out of sample predictive performance. And to do that, in all the SP occupancy model fitting functions, we have these arguments for doing k-fold cross-validation. Okay, so if you want to actually compare the kind of ability of a model to predict at new locations, you can specify these arguments within SPPGOC or whatever function you're using to do that. Okay, and our prediction should wrap up in just a second or so. All right, we're good to go. And I'm just going to kind of run through these last few things just so we have some time for questions. But basically what I'm going to do is I'm going to, if I first look at the results from our prediction object, so this out.pred, remember we predicted at about 42,000 sites, which is pretty good for only running in a couple of minutes. There's going to be kind of three main objects in this prediction object: the z.0 samples, which are the predictions of latent occupancy, so the 0 or 1, whether or not the species is there. These w.0s are the predictions of the spatial random effects at these new locations. And then psi.0 are the predictions of actual occupancy probability at the new locations. Okay, so we can summarize those to generate the maps that we want to do. So what I'm going to do is I'm going to use the apply function to calculate the mean occupancy probability at each location. I'm also going to calculate the standard deviation of the occupancy values at each location as a measure of uncertainty. And I'm also going to do the same thing for the spatial process values, because looking at the resulting predictions of the spatial process can provide some really cool information about kind of the underlying, potentially the underlying covariates that are leading to that residual spatial correlation. Can kind of help you think more critically about your species distribution. Okay, and then this last bit of code, I'm not going to go through in any detail whatsoever. I'm just going to run it. But basically, what it does is it just formats the data for use with ggplot and the stars package for generating a prediction map that we saw before. So I'm going to go ahead and, of course, I have to make my plot window a bit larger in order to generate the file. But you can take a look more closely to see what those look like. And really got to make this big. All right, so if I run that, I should generate a plot. Well, something's gone on with the graphic state just because I'm sharing my screen and the text is a little big. But if you trust me, then if you're running these on your own, then you should see that figure that I showed in the slides of the predicted occupancy probability and the spatial process values. Okay. So that's about it. That's about what we'll go through. I do want to zoom really quick. I'm going to take like 1 minute to mention something with the multi-species models, just to connect it back to what I mentioned in the presentation. So I'm going to zoom over real quick to the multi-species script, the Swiss MHB JSDM. And all I'm going to do is I'm going to load in the data, and I want to talk a bit about the format of the data for fitting a multi-species model in SPI. Okay, so just do this really quickly. If I load that, what we'll see is the data object, this data.swiss MHB. What this is an object formatted for multi-species models. And the main thing I want to mention, the only difference is in how we specify the multi-species detection, non-detection data. Okay, so I think this is the trickiest part, fitting a multi-species model in SPI occupancy, is formatting the detection, non-detection data. Okay, and how we do that connects back to what I mentioned in the slides with those detection, non-detection matrices kind of stacked on top of each other for each species. Okay, so each species has its own site by observation level matrix. Okay, and so what we do in SPI occupancy is you're going to send those into the data list as a three-dimensional object, which you can imagine as those two-dimensional matrices stacked on top of each other. Okay, so this first dimension is going to correspond to total number of species we have. So in this case, we have 145 species. And then our second dimension corresponds to sites, and our third dimension corresponds to those repeat visits. Okay, so that is kind of the only difference in how you would specify the data list object for fitting a multi-species model, is kind of getting the data in this format for a three-dimensional object. And it can be a bit tricky to format this actual object from raw data that you have in CSV or Excel files. I do have an example up on a package website of how to take a kind of messy data set and format it into this format. So do check those out if you're interested in. But I think I'll leave it at that. So I think we can go ahead and start talking about questions as well, if that is helpful.
All right, thank you, Jeff. Fantastic. So if anybody has questions and you haven't posted yet, use the poll everywhere link to post your question, or you can upvote questions that have already been posted. I'm gonna provide the questions to Jeff that have been the most upvoted, so I'm going from top to bottom. The first question is, does the spatial occupancy modeling account for uneven sampling of sites? So sites sampled in 1 year are possibly not sampled every year.
So that's an interesting question. So for uneven sampling over sites, not directly. So basically the spatial random effects are going to be informed by the surveys that you have at that location. So for example, if you surveyed sites 20 times in the western portion of your study region versus 1 or 2 times in the eastern portion, all of those estimates are going to be primarily given based on the sites that have more data. And so it doesn't directly account for uneven balance of those surveys.
However, just to add to that, but because you have the NAs, right, you allow for uneven sampling. It's just that it's going to be more strongly influenced by the sites that have greater number of samples, right?
Yeah, exactly. Thanks for clearing that up, Dennis.
And I'll also jump in and just mention that your posterior predictive distribution should be tighter for those locations that have more data as well, if you're making maps, for example. Potentially, unless there's just more noise in those regions.
And then here, is it possible to make the predictions using raster files for covariance?
So the way the functions are currently written, they don't take directly in a raster object. So what you would have to do is convert that raster object to a matrix or a data frame, and then supply it into the functions. I can't remember off the top of my head, but I think there is a pretty straightforward function that goes from a raster to a matrix object that should allow you to do that pretty quickly.
And then as part of the input data that you give to the model, there are the coordinates of the sites that are sampled. And the question is, are these coordinates, do the coordinates have to be in UTMs? Does it make any difference if we just give latitude and longitude instead of coordinates in UTM?
Right. So you'd want those to be in a projected coordinate system where it's a UTM or something in meters and kilometers. Functions aren't directly designed for the latitude and longitude values. So yeah, you'd want to put them in UTMs.
And so another question here is, are there plans or the possibility to add additional hierarchy into the model? And then the examples here listed in the question are splitting the detection process into availability, perhaps based on survey conditions, and versus detectability, given that the species is available. And so between parentheses, observer skill, habitat variables like vegetation density for visual encounter surveys, things like that.
Yeah, absolutely. I would love to incorporate kind of, you know, multi-scale aspects of availability versus detectability. Those three things I mentioned on kind of the coming soon slides are kind of my top priorities right now. But I do agree that I think it would be very useful to have those aspects in there. So they are on kind of my running list of things to add, but I don't know how quick they would be up there.
The next question is, does the SP occupancy allow for the complementary log-log link? And also if it allows for the inclusion of an offset term that accounts for variation in effort?
Well, those are great questions. So the response to the first one, the clog-log link would be no. So what we use is a logit link, and in particular, we use the logit link with so I mentioned that polyogamma scheme, and that the way we've written up all the functions relies on that scheme, which is specifically for the logit link. And then in terms of the offset term for variation in effort, we don't currently have that in there. So basically, we'd imagine any kind of effort variables you'd kind of throw in on that detection probability portion of the model. And so we don't have functionality for those models that type of model that would just have an offset for effort rather than explicitly modeling detection probability using the kind of hierarchical way that an occupancy model does. But those are great questions.
All right. And there's a question of how much you need to know or understand about the Bayesian framework to be able to use this package. And the person goes on to state that they don't have a strong Bayesian background.
Yeah, that's a fantastic question. So I think with the vignettes and the resources that we provided along with the package, we try to give the kind of basic information we need to get going to fit the models in the package. You will kind of need a basic understanding of you know, when you can rely on results from a Bayesian model, or so assessing convergence and things like that to ensure the model results are valid. But kind of one of the motivations for this was, you know, to be able to fit this kind of Bayesian spatial occupancy model without having to go into the nitty-gritty details of fitting a model using the bugs language or something like that. So you know, I encourage you to check out the vignette materials that we have, and and the resource that we have linked in those for getting more familiar with the Bayesian concepts that you'll need.
Yeah, and Jeff, it seems like you had mentioned maybe the vignette has more information regarding this. So the question here is really focused on data preparation and stating that it's one of the most important and sensitive steps in this kind of modeling. So that it would be really useful if you had a working example script that shows how you can go from regular data frames to the data that, like the data that you showed, the Goldfinch data that's already ready to go to the SP occupancy function.
Yeah, absolutely. So there is a vignette on the package website, and I'm trying to copy it right now so you have it. What it does, I mean, obviously, I could imagine your data being formatted in a million different ways. But what I've done with the vignette that I just sent out in the Zoom link is provide an example of going from a relatively standard formatting for detection on detection data for multiple species, and formatting that for both a single species model as well as a multi-species model. So hopefully that helps. And of course, you can reach out via email if you're having troubles formatting in that way as well. So.
Really fantastic resource. The next question has to do if you can specify splines in the formula that you provide to SP occupancy, something like tilde S elevation or something like that. Is that a possibility currently?
So I will admit I have only played around with that a little bit recently, and it does work. So as long as it is something that can kind of be parsed apart by our model formulas, I think it should work. So I've played around with including some basis splines in the formulas. So it should work. I will mention that I haven't tested it out explicitly for doing that. So you might run into some weird behavior, but you should theoretically be able to include, just like you do with the scale function that I had shown those examples, you should be able to include those additional things in there that can account for, non-linear effects of covariant effects. It's a great question.
And can you talk a little bit more about the n.batch and batch length? So why did you choose 425 when you ran the model? Yeah,
absolutely. That's a great question. I think that's one of the more confusing parts of fitting the models. So as I mentioned, the number of MCMC samples in these spatial models are kind of split up into these two components. So the first thing I would specify would be batch length. Usually my go-to is 25, and I generally don't run into major problems with using 25 for this type of model. So in the vignettes, you'll see that I kind of mentioned I suggest just keeping this at 25 as the kind of standard. And then the n.batch is basically what you would change to increase the number of MCMC samples you're running. Okay, so the higher that is, the more samples that you're running. So in this case, the reason I chose 400 was just because the 10,000 MCMC samples, so 400 times 25, resulted in parameter estimates that were decently close to convergence for that example. But you basically increase the number of batches to a point where you're able to get convergence on your MCMC model. Yeah.
But out of curiosity, why did you guys specify in this way, just not the overall number of iterations? Why batch and batch length?
Yeah, that's a great question. So it comes from, and I'll let Andy elaborate on it too, but so we use an adaptive algorithm so estimation of the spatial parameters, in particular the spatial range parameter, can be a bit tricky. And so what we do is we use this algorithm that kind of tunes it adaptively across the model. And so to do that, you specify these two parameters. So that's kind of why we have this in them.
Yeah, I'll just jump in and mention. So this adaptive Metropolis Hastings algorithm comes out of work by Roberts and Rosenthal, which is, I think, cited in Jeff's package here. And if you just look up adaptive MCMC Metrop within Gibbs by Roberts and Rosenthal, there's a very accessible paper. It's an easy read. You don't have to have a lot of statistical background to sort of understand what they're doing. The general idea is with the number, the 25, it's not a magical number by any regard, but what it does is it essentially samples 25 draws from the posterior distribution. It assesses the acceptance rate of those, if it's accepted or rejected. The Metropolis algorithm is an acceptance rejection algorithm for sampling posterior distributions. If the acceptance rate is too low, then the proposal variance is adjusted, or too high, it's adjusted to essentially hit this target acceptance rate. And so you just need a robust enough sample to get a sense of what the acceptance rate is before that adjustment occurs at the end of each batch. So 25, 30, 50, you can play around with it and see for your given data how quickly essentially the chain acceptance rate is being met. So that you won't break anything if you have a different number. It just may take longer for your chain to converge.
Great. So we're coming to the last couple of questions. Can you talk a bit about troubleshooting the package? For example, if the person did not provide one for the intercept in the prediction matrix, what types of error messages have you guys implemented to account for issues like that?
Yeah, that's a great question. So I think kind of the most problematic part where I've tried to implement a lot of checks is with the data formatting. So I have a bunch of checks off the bat that make sure that the missing values line up, or there's not missing values in improper locations or things like that. And you should see some pretty relatively useful error messages that are printed out compared to some kind of tricky R error message that I see. And then sometimes in particular for that example with the intercept with the prediction, that one might be a bit trickier. That's actually a pretty good idea to implement, maybe a message that says there's not an intercept included in x.0. So that might be a good idea to do for myself in the future. But anyways, you can kind of check, in a case like that, you'd probably see some bizarre predictions. Occupancy probability might be way lower in locations than you think it would be, or way higher. So generally we do try to include error messages that check for, improper formatting and proper specification of prior distributions or initial values or things like that. So hopefully the error messages are kind of catching those locations. But if you do get to a point where there's a really not useful error message, then definitely feel free to reach out, and I would be curious to see where that happened.
Is there a possibility to run each chain in parallel, parallel processing at the moment?
Yeah, that's a great question. So no, at the moment would be the short answer. So basically we have parallelization implemented for kind of two things. So one would be with the spatial models, if you increase the number of threads, that can really help improve runtime of the models. It doesn't do the parallelization across chains, but rather it does it within the chain itself. So the chains are still run in sequence. There is also parallelization that's done with the cross validation as well. The reason I didn't include parallelization in there for the chains at the moment is because kind of led to a lot of potentially things that were messing up with the parallelization within the chain with the spatial models, as well as trying to parallelize across the chains as well. And so what an alternative thing you could do is to kind of do the parallelization separate outside of the package. So just run the chains one by one, do them on different threads. but yeah, not directly within the arguments themselves.
Yeah. So because of time, I have there's only one more question, and that I'm gonna relay soon. The rest of the questions, unfortunately, will not be able to go over. So the last question is, can you describe how to decide when to include traditional random effects that also represent spatial information, like transect ID, forest ID, state, or what have you? If those should be included, or if not, or how to go about deciding in terms of the traditional random effects versus the spatial random effects, if there's going to be an issue of adding both, perhaps.
Yeah, that is a fantastic question, and I should have emphasized, I don't think I mentioned it all, but you can include non-spatial random effects in the model using LME4 syntax. So with that kind of 1 dash, whatever your factor is. In terms of including random effects that have spatial information, I think it can provide you with some really cool information to include those in a model and see if including additional spatial random effects can help improve the model, because that can give you some information on the scale at which the spatial correlation is occurring. So for example, if you fit a model with a spatial random effect across, this example, like forest ID or transect, and you compare that to a model with spatial random effects, the non-traditional spatial random, or the non-traditional random effects model is a more parsimonious model, right? It's more simple. And if the spatial autocorrelation is occurring at that scale, you might see that model does just as well as the spatial random effects model. Okay, so I would suggest it could be a pretty useful model comparison exercise to kind of do those comparisons when you're fitting models, if you do hypothesize that spatial autocorrelation is occurring at that scale. But again, the spatial random effects component will allow for kind of a more fine scale spatial variability that may occur that's not directly accounted for with those transects or forest stands or something like that.
Yeah, I just might add, just reiterate what Jeff said. If you think you have multiple sort of scales of influence going on in your data, you may include something broad scale like ecoregion or watershed, Occ units or something like that, or management, to pick up on these really broader scale impacts, or maybe climate zones, and then leave the spatial random effects in there to sort of soak up any local scale variation within those units. So certainly able to do that.
Great. All right. So thank you very much, Jeff and Andy. This was really great.